Hello, very good evening. Kindly wait, we will start our session at 6 p.m. Let other students join, okay? I hope my voice is audible to you. So we will start our session at 6 p.m. Till then, kindly wait. Good evening to the students who have joined. Can you let me know if my voice is audible to you? And also, please let me know if you can see the screen, the presentation, if it is visible. The exam pattern, I believe it is already mentioned in your SWAM portal, right? If you go through the SWAM portal, you will find that there is the medicinal chemistry page itself. The announcement there, the exam pattern is given. So just let me take you to that portion. Yeah, this is the exam format that is given already. Okay, so 1 second. Yeah. So is it visible, the screen? Okay, let me show you. So, if you see that there is this particular thing, okay. So this is the exam pattern for the medicinal chemistry course that is already given. Okay, so here you will see there is a type of examination. If you click on the list, you will find that for our medicinal chemistry, it is the CBT or the computer-based examination. Okay. So in case of the computer-based examination, so the questions will be on computer and answers you have to and enter on the computer. And the type of questions may include the multiple choice, fill in the blanks, and essay type answers. Okay. So this will be the pattern of your examination. That's all that can be. I mean, I can let you know right now. Otherwise, and there will be a on-screen computer. The demo you can use this link to use the calculator before your examination to get a idea how to use this calculator. So that is this. If you can go through this particular thing and you can check. Okay. So that will be your exam for pattern and formation. Mostly you will have the MCQs and fill in the blanks or essay type examination, but all the thing you have to enter through the computer. Okay, because it is a computer-based examination. For any other query, you can directly email to the NPTEL, the authority. They can let you know about all these things, okay? If you need more details around it. That's what I know, and that's what I can convey you about the examination pattern. So, good evening to the students who have joined. I hope my voice is audible, and also I hope that you can see the presentation, right? Just let me know one time that if the presentation is visible to you or not. The PowerPoint presentation, is it visible to you? Please let me know. Okay, I assume that the presentation is visible to you. So today it is our 12th class, and after that we will have one more class which is on tomorrow. I think you will receive the notification regarding tomorrow's class. That will be the last class of our session. That is a summary extra class. Okay, and otherwise today it is the last no, regular classes because it is a week 12 live session. Please wait till it is 6 p.m. and then we will start the session. Kindly let me know if there is any issue with the audio, video, or presentation. Please let me know. Okay. Okay, so I think I should start the session. I hope my voice is audible and you can see the presentation and the screen clearly. So, very good evening to the students who have joined. My name is Mousumi Deb and I'm a PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL course of Medicinal Chemistry, and today it is our week 12 live session. So, till now, we have completed for this course 11 classes till now, and today it is the 12th live session. So our course medicinal chemistry is a 12-week course, and today it is the last class of our this particular course. Okay, so during this medicinal chemistry course, we have already learned in our couple of past weeks, we have learned about the basics of medicinal chemistry, followed by the proteins, the enzymes, the inhibition mechanisms, and also then we have completed all the things like the ADME, absorption, metabolism, drug administration part, also the phase 1, phase 2 metabolism, drug designing strategy. Then we moved forward towards therapeutic agents like antibacterial, antiviral, anti-cancer agents, and so on. So this is our last class, the last regular class of this course, okay, this week 12 live session. And after that, we will not have any this regular classes till now, okay, because after that, we will be, you will be having your examination soon. And but before that, tomorrow I will have another class, extra session that will be from 4 p.m. Okay, from 4 to 6, we will have an extra session. So in extra session, I will just randomly show you some questions and from different parts of your course, and we will discuss it. Some things which I couldn't discuss earlier that I will discuss tomorrow. And regarding the extra class, you will be notified by email regarding the schedule. Okay. So I think I have already mentioned a couple of times where to find all the materials. So you have to go to the SWAYAM portal, the medicinal chemistry. On the announcement page, you will find there is a left panel, and on that of the left panel you will find this problem solving session January 2026. If you click on that problem solving session recording, you will be redirected to a page which looks like this contain all the date wise, you will find all the recordings of our classes whatever we have conducted till now, and also you will find a presentation link with the Google Drive link. This Google Drive link, this has all the presentations whatever we have completed, that you will find. Okay, so you can go through the material, you can download, and you can go through the videos and the presentations from that portion. So today it is our week 12 live session. So before starting any session, just I mentioned few things that will be same. That the session is live and I have muted your microphones and cameras so that there is no issue with the recording, because I am recording the session, and the recording it will be shared to you after class along with the presentation whatever I am showing. Okay, so these two things will be I will share to you after the session today itself, or by tomorrow morning it you will find it on that particular Google Sheet. So if you have any doubt or any query, you can post it through the chat box, okay? And if after class you have the doubts, and you can drop an email to me, I will share my email ID after at the end of the session, okay? And so let us go through the material. Let us go through the first question of the first today's session. Let's see first what are the topics. So mainly we will discuss today two different topics. One is the QSAR. Okay, and one is and the another is the drug resistant and synergy. So SAR, what is SAR? I think I have discussed it in our fifth or sixth class. We have detailed discussed that SAR. That SAR or structure activity relationship study is that when you make small changes in the structure, how it activity varies. Okay, for example, you have a particular, you are developing a series of a particular therapeutic agent, let's say for antibacterial purposes, and let's say you have made a series of 20 molecules, okay, and each molecules are structurally related to each other, only different, the differences are some group or another. For example, let's say one have a phenyl group at the para position of a particular side chain, but another has a methoxy group, or another has a methyl group. So that's how we change the molecules by little by some structural features, like by adding free electron withdrawing or electron donating group, or removing some part, or adding some hydrogen bond donor acceptor, or elongating the chain, or removing some it double bond, or adding some double bond. So the structural changes that has been that basically that we do, that is known as the structure activity relationship studies. Okay, this is how the small changes in structure varies the activity of a particular compound. But QSR is something different. QSR, or the quantitative SARs, that is something different, that a topic that we will discuss today, and we will discuss about the drug resistant and synergy. Okay, So we have discussed a lot about various drugs, various therapeutic, but resistance is another topic that we are yet to discuss. So these two things we will be doing in today's class. Okay. So now let me show you the first question of today's session. So which of the following statements is false regarding the QSAR? Your options are: number A, for the QSAR study, the compound studied need not to be structurally related. Number B, correct testing procedures must be used. Number C, in vitro tests carried out on isolated enzymes are relevant. And number D, none of the above. Which of the following statements is false regarding QSAR? Number A, for QSAR study, the compound studied need not to be structurally related. Number B, correct testing procedures must be followed. Number C, in vitro tests carried out on isolated enzymes are relevant. And number D, none of the above. So, which of the following statements regarding QSAR is false? Let me know A, B, C, or D. Which of the following statement regarding QSAR is false? Like every day, again, I will say that whenever there is a true, false, or correct, incorrect type of question, take your time and read the questions and options very carefully before answering. Because, as I have mentioned earlier, also your examination will also be CBT. So be careful whenever answering a multiple choice question or multiples or the questions which have multiple correct answers. Read of the options carefully, think, and then let me know. I will wait for a minute or two and then I will check the chat box and check your answers. After that, I will show you the answer and we will discuss this particular QSR in detail. Which of the following statements regarding QSR is false? Number A, for the QSR study, the compound studied need not to be structurally related. Number B, correct testing procedures must be used. Number C, in vitro tests carried out on isolated enzymes are relevant. Number D, none of the above. Let me check the chat box. Let's see if there is any answer for our problem number one. Okay, number C is the given answer. Let me check if number C is the correct answer or is there anything else. So, the correct answer will be the statement which is false will be it is number A. Okay, for QSR study, the compound studies need not to be structurally related. This statement is false. So the QSR approach attempts to identify and quantify the physicochemical properties of a drug and to see whether any of these properties has an effect on the drug's biological activity. If such a relationship holds true, an equation can be drawn up which quantifies the relationship and allows the medicinal chemist to say with some confidence that the property or properties has an important role in the pharmacokinetics or mechanisms of action of the drug. It also allows the medicinal chemist some level of prediction. By quantifying physicochemical properties, it should be possible to calculate in advance what the biological activity of a novel analog might be. There are two advantages of this. Firstly, it allows the medicinal chemist to target efforts on analogs which should have improved activity and thus cut down the number of analogs that have to be made. Secondly, if the analog is discovered which does not fit to the equation, it implies that some other feature is important and provides a lead for the further development. So, what are these physicochemical features which have mentioned? Is essentially they could have been any structural, physical, or chemical property of the drug. Clearly, any drug will have large number of such properties, and it would be a Herculean task to quantify and relate them all to the biological activity at the same time. A simple, more practical approach is to consider one or two physicochemical properties of the drug to vary these while attempting to keep the other properties constant. This is not as simple as it sounds when it is not always possible to vary one property without affecting another. Nevertheless, this has to be taken numerous examples where this approach has worked. It is important that QSR method is used properly and in relevant situations. Firstly, the compound studies must be structurally related. So that's why the statement number A is false, which says that for the QSR study, the compound studied need not to be structurally related. No, the compound studied must be structurally related, act at the same target, and have some mechanism of action. Secondly, it is crucial that the correct testing procedures are used. In vitro tests carried out on isolated enzymes are relevant for a QSR study as the activities measured or for different inhibitors are related directly to how each compound binds to the active site. In vivo tests carried out to measure the physiological effects of the enzyme inhibitors are not valid. However, as both pharmacodynamic and pharmacokinetics factors come to play, this makes it possible to derive a sensible QSR equation. Okay. So the statement number A, which says that for the QSR study, compound studied need not to be structurally related, this is false. Okay. So is it clear, or do I need to repeat anything? Okay. If it is clear, then I will move to the next question. Okay. So statement number one, which says that, so the main thing that you have to remember is this part. Okay, that the compounds studied must to be structurally related to act at the same target and have same mechanism of action. Okay, because you cannot compare one topoisomerase inhibitor with an let's say with an alkylating agent, right? You have to compare an alkylating agent with an alkylating agent and a topoisomerase inhibitor with a topoisomerase inhibitor, and because they must have same the mechanism of action. Secondly, it is very critical to correct testing procedures are used, and in vitro tests carried out on isolated enzymes are relevant for a QSAR study as the activities measured for different inhibitors are related directly to how the compound binds to the active site. In vivo tests carried out measured are not valid. Okay, this is important that the in vivo test carried out to measure the physiological effects of enzyme inhibitor are not valid as both pharmacodynamic and pharmacokinetics factors come to a play. Okay, so this is impossible to derive a sensible QSR equation from the in vivo data, because the in vivo data not only it depends on the effect of the particular drug on a enzyme, but also it affects on the pharmacokinetics and pharmacodynamics of that particular drug in the body of the patient, right? So that is why in vivo data is not relevant, but in vitro data are relevant because that is performed on a isolated enzyme. Okay. Now I will go to the next problem. Problem number two says which of the following is not a QSR parameter? Your options are number A, hydrophilicity, hydrophobicity, number B, hydrophilicity, number C, steric, and number D, electronic. Which of the following is not a QSR parameter? Hydrophobicity, hydrophilicity, steric, or electronic? Let me know A, B, C, or D. Which of the following will be the correct answer? Which of the following is not a QSR parameter? I will wait for 2 minutes, then I will check the chat box. Okay, A, B, C, or D, let me know which of the following is not a QSR parameter. Hydrophobicity, hydrophilicity, steric, or electronic. Which of the following is not a QSR parameter? Okay, let me check the chat box. Okay, number B I have received as answer. Two number B and one number D I have received as answer. Okay, let me now, I have to just check which of the following will be the correct answer. Okay, okay, so the correct answer will be number B, hydrophilicity. Hydrophilicity is not the QSAR parameter. So what is the QSAR? Already I have discussed it a little bit. The quantitative structure activity relationship or QSAR is a mathematical relationship which correlates the measurable or calculable molecular properties to some specific biological activity in terms of equation. QSAR attempts to find consistent relationship between biological activity and molecular properties so that these rules can be used to evaluate the activity of new compounds. Understanding QSAR regarding the electronic effects, steric effects, and lipophilicity. So the various parameters used in QSAR studies are the hydrophobicity, partition coefficient, pi substituent constant, steric parameters, Stubbs constant, Verloop steric parameter, and electronic parameter like Hammett constant and dipole moment. So physicochemical properties, many physical structural chemical properties have been studied by QSR approach, but the most common are the hydrophobic, electronic, and steric properties. Okay, so these three are the most common properties that has been studied in the QSR studies. This is because it is possible to quantify these effects. Hydrophobic properties can be easily quantified for a complete molecule or for one individual substituent. However, it is more difficult to quantify electronic and steric properties for complete molecule, and this is only really feasible for the individual substituent. Consequently, QSR studies on variety of totally different structures are relatively rare and are limited to studies on hydrophobicity. It is more common to find the QSR studies being carried out on compounds of the same general structure where substituents on aromatic rings or accessible functional groups are varied. The QSR study is then considers how the hydrophobic, electronic, and steric properties of substituent affect the biological activity. The three most studied physicochemical properties are now considered. So I will discuss it in detail later. Detailed in this physicochemical properties that are studied in the QSR parameter. So remember, the hydrophobicity, steric, and electronic properties are the properties that are most studied in the in our QSR, and there are several constant and equation that are used to quantify these things like partition coefficient, pi substituent coefficient in case of hydrophobicity, steric parameters like Taft constant, Verloop steric constant, and Hammett constant, dipole moment in case of electronic. Now I will go to the next problem. Problem number 3 says, which of the following statements is true? Number A, hydrophilic compounds have a high value of P. Number B, hydrophobic compounds have a high values of P. Number C, acidic compounds have a high value of P. Number D, basic compounds have a high value of P. Here P defines as the partition coefficient. Okay. So in case of your examination, not all the time you will be given the complete versions of every, terms. You will be given the shorthand notations and acronyms also. So be careful and be mindful while you are preparing for your examination. Which of the following statement is true? Number A, hydrophilic compounds have a high value of P. Number B, hydrophobic compounds have a high value of P. Number C, acidic compounds have a high value of P. And number D, basic compounds have a high value of P. Let me wait for a minute, then I will check the chat box, okay? Which of the following statements is true? Let me check the chat box. Okay. Number A, number A. So I have received two answers and both are saying that number A is the correct answer. Okay, let me check if number A is the correct answer or not. So the correct answer will be, it is number B. Okay. Hydrophobic compounds have the high value of P or partition coefficient. Now let us understand what is the partition coefficient and how it matters, why it is very important in case of our QSAR studies. So the partition coefficient P is the most widely used parameter in QSAR because it directly relates to molecules lipophilicity or fat solubility to its ability to reach the target within the body. So now what is the partition coefficient? It is the ratio of the drug's concentration in the non-polar solvent, usually 1 octanol, versus a polar solvent, that is usually water. Okay. And at equilibrium. Because these values can span several orders of magnitude, we typically express them as the logarithm. So log P value equal to log of the concentration of a particular drug in octanol or polar medium divided by log of divided by the concentration of the water of that particular drug. Okay, so high log P value, the drug is lipophilic or fat loving or hydrophilic, we can say. Okay, it crosses the cell membrane easily but have poor water solubility. And lower log P is the drug is hydrophilic, the water loving. It dissolves in the blood and struggles to cross the it dissolves well in the blood but struggles to cross the fatty layers like the blood-brain barrier. Okay, so now the hydrophilic compounds, they have high P value. This is not true. Hydrophobic compounds have high P value. This is right. Acidic compounds and basic compounds, we cannot say for sure that they will have the high value or not. Okay. Generally, the acidic and basic compounds are water soluble. In most of the cases, they have good water solubility, so they will not have a very high log P value. So why 1 octanol is used as a polar medium to calculate this log P value? So octanol is chosen as the standard organic solvent for two main reasons. One is the biological mimicry. It has long hydrocarbon chain and polar hydroxyl group makes it structurally similar to the phospholipids that make the human cell membrane. And number two is the hydration. Octanol can dissolve small amount of water, mimicking the hydrated environment of the real biological membrane. The bilinear relationship Hans analysis in QSAR, the relationship between log P and biological activity is rarely a straight line. Instead, it follows a parabolic curve or U-shaped curve which is upside down. The linear range initially a log P increases, activity increases because the drug enters to the membrane more easily. Optimum log P or log P0, there is an ideal lipophilicity where the drug is perfectly balanced with the being soluble enough to travel through the blood and lipophilic enough to enter cell. And drop off if the log P value is too high, the drug gets trapped in the first fatty tissue and it encounters like body fat and never reaches to the target receptor. So these are the bilinear relationship or Hans analysis regarding the QSAR and our log P values. Okay. So the correct answer was number B, not number A. So is it clear? What is the log? What is a P? What is a partition coefficient? And why the lipophilic and these things works like? So number A was not a correct answer. Okay, but it's fine. So because as I already mentioned that the answering during the class is important. Even if your answer is wrong, it is fine. Okay, but at least try to answer regularly. So now I will go to the next problem. So problem number 4 says the correct equation to find out the substituent hydrophobicity constant pi x equal to your options are number A log px minus log pH, number B log px divided by log pH, number C log px plus log pH, and number D log px multiplied by log pH. Which will be the correct equation to find out the substituent hydrophobicity constant pi x? So this pi x is very important. This is known as a substituent hydrophobicity constant. Okay, this term. Why it is important, I will be discussing in a couple of minutes. But first, you have to identify this substituent, which of the following is the correct depletion or the correct representation of this substituent constant pi x. Number A log px minus log pH, number B log px divided by log pH, number C log px plus log pH, and number D log px into log pH. Let me know A, B, C, or D, which of the following will be the correct answer. The correct equation to find out the substituent hydrophobicity constant or pi x will be, let me check the chat box now. Let's see if there is any answer. Okay. Number A, number A, number A. So I have received three answers and all are saying that number A is the correct one. Okay, let me find out if number A is the correct one or not. So the correct answer is very good. It is number A, log px minus log pH is the correct definition of this substituent hydrophobicity constant or pi x. So now let us discuss or let us understand what is this substituent hydrophobicity constant. Let me highlight the important parts. So we have seen how the hydrophobicity of a compound can be quantified using the partition coefficient. So in our last slide, I have mentioned that hydrophobicity and partition coefficient are deeply correlated. In order to get this partition coefficient P, we have to measure it experimentally, and this means that we have to synthesize the compounds. It would be much better if you could calculate the P theoretically and to decide the advance whether the compound is worth synthesizing. QSR would then allow us to target the most promising looking structure. For example, if we were planning to synthesize a range of barbiturate structures, we could calculate the log P values for them all and concentrate on the structures which are the log P values closest to the optimum log P0 of the barbiturates. Okay. So the partition coefficient can be calculated by knowing the contribution that various substituents make to the hydrophobicity. This contribution is known as the substituent hydrophobicity constant. Okay. Various substituents for a particular compound, various substituents or various functional groups that will contribute something to the hydrophobicity of the whole molecule, and these particular contributions are known as the substituent hydrophobicity constant or pi. This is a measure of how hydrophobic a substituent is relative to hydrogen. The values can be obtained by as follows: the partition coefficient measured experimentally for a standard compound such as benzene with or without substituent X, and the hydrophobicity constant pi x of which the substituent is obtained by using this equation. This pi x equal to log px minus log pH, where pH is the partition coefficient for the standard molecule and px is the partition coefficient of the standard compound with the substituent. The positive value of pi indicates that the substituent is more hydrophobic than hydrogen, and a negative value indicates that the substituent is less hydrophobic. The pi value, so this is important, positive pi value indicates that the substituent is more hydrophobic than hydrogen, and a negative pi value indicates that the substituent is less hydrophobic. Okay, and the pi values range of substitution, there is a table that I will show you. Here is the table. Okay, so these pi values are characteristic for the substituent and can be used to calculate how the partition coefficient of a drug would be affected if these substituents were present. The p value for the lead compound would have been measured experimentally, but once this is known, p value of the analogues can be calculated quite simply. So various pi value pi for the range of substituent, the group pi aliphatic and aromatic substituent. So if there is these substituents are aliphatic, the for CH3 it is 0.50, TB the tertiary butane it is 1.68, OH is minus 1.16, for OCH3 0.47, CF3 is 1.07, chlorine is 0.39, bromine is 0.6, and fluorine is minus 7. So if you check, if you see that in case of the halogen containing, in case of the aliphatic one, the fluorine has a negative value, means fluorine is more hydrophilic in case of the aliphatic than hydrogen, but chlorine bromines are more hydrophobic along with CF3, but in aliphatic all are more hydrophobic than fluorine hydrogens. Okay, but OH, OMe, these are the hydrophilic, and this distributor and methyl are also hydrophobic. So the values you do not need to memorize. Okay, whenever you will, if there is any question where you have to calculate the pi x, the values will be given for the substituent. But just, I mean, remember that, or just try to understand why a particular value is positive or negative. So that will help you to understand that particular point. Okay. Now I will go to the next problem. Problem number 5 says, given the following log P values, what is the log P value of metachlorobenzene? So this is the values are given. Okay, for benzene, the log P value is 2.13. For chlorobenzene, it is 2.84. For the benzamide, the value is log P is 0.64. Now you have to identify what is the log P value of metachlorobenzamide. Your options are number A 1.97, number B 2.01, number C 1.35, number D 2.29. What is the log P value of metachlorobenzamide? Number A 1.97, number B 2.01, number C 1.35, and number D 2.29. The log P values of benzene, chlorobenzene, benzamide are given as mentioned in this figure. You have to calculate the value of metachlorobenzamide. Try to answer the question. Whatever I have shown you in the last slide, you have to use that particular equation. If you use that equation, it is very easy question actually. It is very easy mathematical question. Okay, I will wait for 2 minutes, then I will check the chat box. Okay, take a pen and paper and try to solve this question. This kind of small mathematical questions will be given to you, so it is better that for you to practice this kind of questions. Given the following log P values, try to calculate the log P value of metachlorobenzamide. After a minute, I will check the chat box. Just you have to use the particular equation that I have mentioned in our last slide. If you use it, you will easily be able to answer this question. Let me check the chat box. Let's see if there is any answer. Okay, I have received a couple of answers. Okay, only two, number C and number D. So two different answers I have received, number C and number D. Fine, let me now check which of the following will be the correct answer. So the correct answer is number C, 1.35. Very good. There was an answer, I think number C. So now let us find out how to calculate. So this is the particular table that I have mentioned in our last slide, right? This particular table, the particular values of the pi range. Okay. So if you do not remember, so but these values are given. So now log P of metachlorobenzamide, what it will be? So the for this compound, the basic structure is benzene, and the substituent you have one chloro and one CONH2, right? So the log P value of metachlorobenzamide will be equal to log P value of benzene plus the pi value of chlorine and pi value of CONH2. Now you have to figure out what are the pi values of chlorine and CONH2. In case of the pi value of chlorine, that particular that it will be for this chlorobenzene it is 2.84 minus the value of benzene means 0.71, and in case of the CONH2 it is the value of benzamide which is 0.64 minus the value of benzene which is 2.13, that is minus 1.49. right? So then log P value of this particular metachlorobenzamide will be log P value of benzene 2.13 plus the log P the pi value of chlorine, which is 0.71, and plus the pi value of CONH2, that is minus 1.49, that is 1.35. That is the value of the metachlorobenzamide. So these type of questions can be given to you. Different kind of substituents can be used. Different types of the values can generate. So that's how you have to solve. So it is actually very easy type of question. So you do not need to remember the chart, but the from the given values you have to figure out what will be the correct answer. Okay. So yes, number C was the correct answer. Now I will go to the next problem. Problem number 6 says match the following tough steric factor values with the substituent. Your options are number A. So you have to match the values with the. So this is the tough steric factor values, not the last one what we have done earlier. So this is a different thing. Okay. So your values are number A. Number A is the hydrogen, number B methyl, number C isobutyl, and number D cyclopentyl. And the values are number 1 1.24. Number 2, 0 minus 0.93, number 3 minus 0.51, and number 4 is a 0. So you have to match option number A, 1, A with 1, B with B3, C4, D2, number B, A4, B1, C3, D2, number C, A4, B2, C1, D3, and number D, A1, B4, C2, D3. Which of the following will be the correct answer? Which of the following is the correct matching for the tough steric factor with the value with this substituent? I will wait for a minute or two and then I will check the chat box. Okay. Look all the options carefully, do the matching and then let me know. Match the following tough steric factor values with the substituent. Hydrogen, methyl, isobutyl, cyclopentyls are given. You have to identify which substituent will have which value. Let me check the chat box. Let's see if there is any answer. Okay, number D I have received as an answer. Okay, only one answer I have received, that is number D. Now let me check if number D is the correct answer or not. So the correct answer is very good. Number D. A is with 1, then B with 4, C with 2, and D with 3. So now let us understand what are the steric factors and what is this tough steric factor. Let me highlight the important thing. Okay, let me just use the laser pointer. So the bulk, the size and shape of the drug will influence how easily it can approach and interact with the binding site. Okay, so this is the hydrolysis of a normal hydrolysis of an particular ester. Okay, so this is the ester. After hydrolysis, it will form this acid and we will have methanol. Why? Because it is a methyl ester. Okay. So a bulky substituent may act like a shield and hinder the ideal interaction between a drug and its binding site. Alternatively, a bulky substituent may help to orientate a drug properly for a maximum binding and increase the activity. Steric properties are more difficult to quantify than the hydrophobic and electronic properties. Several methods have been tried and of which three are described here. It is highly unlikely that the drug's biological activity will be affected by the steric factors alone, but these factors are frequently found in the Hand's equation, which is 18.3. So that we will discuss later. So what is the tough steric factor? Attempts have been made to quantify the steric features of a substituent by using the tough steric factor or ES. The value of ES can be obtained by this comparing the rates of the hydrolysis of the aliphatic ester against the standard ester in the acidic condition. Thus, this ES equal to log Kx minus log Ko, where Kx represent the rate of hydrolysis of the aliphatic ester bearing a substituent X, and Ko is the represents the K0 is the representative of the rate of the hydrolysis of a reference ester. The substituents that can be studied by this method are restricted with those with interacted sterically with the tetrahedral transition state with the reaction, not by the resonance or initial hydrogen bonding. For example, unsaturated substituents which are conjugated to the ester cannot be measured by this procedure. Example of ES are given in this table. Okay, so not the where the reference that the reference ester is X. Okay, that is why for methanol, for methyl the value will be 0 because it is the reference ester. Okay, so substrates such as hydrogen and fluorine, which are smaller than the methyl, have the faster rate of hydrolysis. That's why ES is positive. Substrate which is a larger methyl group that reduces the rate of the hydrolysis. Okay, that is why the ES is negative. So, as you can see, the 0 is for methyl. Okay, the positive is for hydrogen because the rate of the hydrolysis will be faster because hydrogen is less bulkier than methyl. And for isobutyl cyclopentyl, that also will be more because these are the more bulkier. The disadvantage of ES values is that they are measure of an intermolecular steric effect, whereas the drug interact with the target binding site with an intermolecular manner. For example, consider the ES values of isopropyl, N-propyl, and N-butyl. The ES values of the branched isopropyl is significantly greater than the N-propyl group because it is linear since the bulk of the substance is closer to the reaction center. Expanding the alkyl into the N-propyl to N-butyl has little effect of the ES. The larger N-butyl group is extended away from the reaction center, so it has a little additional steric effect on the hydrolysis. As a result, ES values for the N-butyl group undervalues the steric effect, which this group might have been present of the drug approaching to the binding site. Okay. So this is all about the Taft equation, and this is the value. So very few values are given. If possible, try to remember few values like for methyl, hydride, fluoride. The values are important, and other groups which are bulkier than the methyl all will have a negative value. So, okay, the more bulkier the more bulkier group is, the more negative the value will be. So now I will go to the next problem. Problem number 7 says, which of the following statements regarding standard deviation is true? Your options are: number A, standard deviation is a measure that is used to quantify the variation of the dispersion of a data set. Number B, a low is a standard deviation indicates the data points are spread over a wide range of values. A high standard deviation indicates the data points are close to the mean of the data set. Number D, none of the above. Which of the following statement regarding the standard deviation is true? Number A, standard deviation is the measure that is used to quantify the variation or dispersion of a data set. Number B, a low standard deviation indicates that the data points are spread over the wide range of the values. Number C, a high standard deviation indicates that the data points are close to the mean of the data set. And number D, none of the above. Which will be the correct answer for problem number 7? Which statement will be true? Let me know. I will wait after a minute. I will wait for a minute and then I will check the chat box. Which of the following statements regarding standard deviation is true? Number A, standard deviation is a measure that is used to quantify the variation or dispersion of a data set. Number B, a low standard deviation indicates that the data points are spread over a wide range of values. Number C, a high standard deviation indicates that the data points are close to the mean of the data set. And number D, none of the above. Let me check the chat box. Let's see if I have received any answer. Okay, so number A, and number B. So I have received three answers. One is number B and other two are number A. Okay, let me check which of the following will be the correct answer for our problem number 7. So the correct answer will be number A. Standard deviation is a measure that is used to quantify the variation or dispersion of the data set. This statement is true. Okay. So, the standard deviation or SD or S is a measure that is used to quantify the amount of variation or dispersion of a data set values. A low standard deviation indicates that the data points are tend to close to the mean. Also, it is called the expected value. So, this statement number B, which says that the low SD indicates that the data points are spread over a wide range of values. This is not true because the low SD values indicates that the data points are tend to close to the mean. While a high standard deviation value means the data points are spread over the wide range of values. So number B and C, the statement both are false, and number D does not account. So number A is the correct answer. Okay, other statistical tests are also conducted to validate this trend. So this is the what is the formula that is used to calculate the standard deviation. So n is the number of data points, xi is the each of values of the data, and x bar is the mean of the xi. Okay, so the value, the standard deviation is the you have to find out this xi minus x, the whole square, and you have to sum of this and divide it by n minus 1, and you have to root of this. So this is the curve, a normal distribution curve it looks like. And so in this position from the this 1 to minus 1, you will have this 99.7% of data points. So, and then from minus 2 to 2, you will have 95, and minus 3 to 3, you have the okay, sorry, it's from minus 3 to 3, you will have this 99.7%. Now from minus 2 to 2, you will have 95, and 1 to 1, you will have 68%. Okay, so this is the graph looks like. And you just try to remember the, equation. Some question can be given for using that you to calculate the some values using this particular formula. So if it is given, you should be able to answer the question correctly. Okay. So a statistical, in short, standard deviation is the statistic that measures the dispersion of a data set relative to its mean and is calculated by the square root of its variance. Okay, so this complete value, it is known as the variance, and the square root of variance is the particular standard deviation. Okay, and this is the mean, and this is the standard deviation. So I hope it is clear. Number A was the correct answer, and it is you have correctly mentioned it in the chat box. Now I will go to the next problem. Problem number 8 says, which of the following statements regarding Craig's plots is false? Your options are: number 8 is a plot where x-axis indicates the value of pi factor and y-axis indicates the value of the sigma factor. Number B, this plot shows the close relationship between sigma and pi. Number C, from this plot it is possible to tell whether which substituent have positive pi and the sigma values and which substituent have a negative one. And number D, from this plot the substituents having similar pi and similar sigma values can be observed. Which of the following statements regarding the Craig's plots is false? Again, I will repeat. Number A, it is a plot where the pi axis indicates the value of pi, x axis indicates the value of the pi factor, and y axis indicates the value of the sigma factor. Number B, the plot shows close relationship between pi and sigma. Number C, from this plot, it is possible to tell which substituents have a positive pi and sigma values and which substituent have a negative one. And number D, from this plot, the substituents having similar pi values and similar sigma values can be observed. Which of the following statements regarding Craig's plot is false? Again, I am saying whenever there is a true-false kind of question, read all the options carefully and then answer. Okay. I will check your answers after a minute. Till then, try and read the options carefully and let me know. A, B, C, or D, which of the following will be the correct answer for our problem number 8. Which of the following statements regarding Craig flaws is false? Let me check the chat box. Okay, I think I have mentioned it in the beginning of our today's class. So, and it is also mentioned in your particular medicinal chemistry, I mean the page, the announcement page. So, if you go through this particular announcement page, you will find there is a exam format that is given for your April 2026 examination. It is clearly mentioned that so if you are from this list, you can check for this course. So I have already checked. So I think this course, so if you click on this, just let me show you. So your course is the medicinal chemistry. Let me go to the course first. Okay. Yeah, so our course is medicinal chemistry. This medicinal chemistry course, you will have a online computer-based examination. Okay, so for online computer-based examination, the questions will be on computer and answer you have to enter on computer, and it will be like multiple choice question, fill in the blanks question, and essay type question. Okay, so mostly not very, I mean, theoretical questions will be asked where you have to write very long, large paragraphs. Okay, it will be like small answer type questions that you can easily answer by typing it on, and also some little paragraphs can be asked, or mostly it will be like MCQs and MSQs and all these things. Okay, so that will be the pattern. Okay, so I think there is no answer for the problem number 8. Okay, let me show you the answer and let us discuss about the Craig's plot. So basically, now the correct answer is number B. The statement which is false, it says that this plot shows the close relationship between pi and sigma. This statement is false. Okay. So the Craig plot, although the tables of pi and sigma factors are readily available for a large range of substituents, it is often easier to visualize the relative properties of the different substituent by considering a plot where the y-axis is the value of sigma and the x-axis is the value of pi. Such plot is known as the Craig's plot. So this is this is the Craig's plots. Okay, this is how the Craig's plots look. This example, here this is for the sigma and pi factors for the para-aromatic substituent. There are several advantages of the Craig plot. The plot show clearly that there is no overall relationship between pi and sigma. So this is important that from that this is an important thing that we can identify from this plot that there is no overall relationship between pi and sigma. Okay, various substituents are scattered all over the four quadrant of the plot. It is possible to tell at a glance which substituent will have a positive pi and sigma parameter, and which substituent have a negative pi and sigma parameter, and which substituent will have one positive and one negative. So this is the thing. Here in this quadrant, we will have both the positives here. Sigma is positive, pi is negative. Here both are the negative, and here sigma is negative and pi is positive, right? So this is easy to see whether the substituent have a similar pi values. For example, the ethyl, bromo, trifluoromethyl, trifluoromethyl sulfonyl groups, all the approximately on the same vertical line of the plot. And okay, so in this these groups can be interchangeable on the drugs. Where the principal factor affecting the biological activity is the pi factor. Similarly, groups which form a horizontal line can be identified with the electronic or similar sigma values. Okay. So this is the how you have to interpret this Craig plot, and why which are the data that can be derived from this Craig plot that is given. Okay. Now I will go to the next problem. Problem number 9 says which of the following statement regarding the drug salmazole is false? Your options are: number A, the drug is free from the CNS side effect. Number B, the log P value is 1.17. Number C, the 4-OMe group was replaced by the 4-SOMe group. And number D, the log P value of the drug is 2.59. Which of the following statements regarding the drug salmazole is false? Number A, the drug is free from the CNS side effect. Number B, the log P value of the drug is 1.17. Number C, the 4-OMe group was replaced by 4-SOMe group. And number D, the log P value of the drug is 2.59. Which of the following statement regarding the drug salmazole is false? Let me know A, B, C or D. Again, I will wait for a minute and then I will check the chat box. Which of the following statement regarding the drug salmazole is false? Let me check the chat box. Okay, I have received one and two answers. One is number C and one is number B. Okay, let me check which of the following answer is correct, number C or number B, or there is any other answer. So the correct answer will be number D. Okay, the log P value of the drug is 2.59. This statement is false. So the cardiotonic agent one. Okay, now one second. Just let me figure out. So this is a cardiotonic agent. Okay. So when R is OMe, it is a one kind of agent, and when R is SOMe, it is the salmazole. So the cardiotonic agent one was found to produce the bright visions in some patient, which implied that it entered the central nervous system or CNS. The supposed to have because the fact the log P value was 2.59. So when R equal to OMe, the drug has a very high value of the log P value, which is 2.59. In order to prevent the drug entering the CNS, the 4-OMe group was replaced by 4-SOMe group. This particular group approximately same size of the methoxy but more hydrophilic, and the log P value is now drug salmazole was found to be 1.17. The drug is now too hydrophilic to enter the CNS and free from CNS. So the drug, the first drug, which has the R as OMe, this has a log P value of 2.59. But the higher the log P value, we know the more hydrophobic the drug is, and because of the high hydrophobicity, the drug was able to cross the blood-brain barrier, and it was creating some issues with the central nervous system. That is why this OMe is replaced by SOMe, which has a more or less similar structure like the OMe, but it is more hydrophilic. That is why the log P value, it is lowered to 1.17, and now the drug is too hydrophilic to reach the CNS, and the drug was free from the CNS side effect. So regard the statements now regarding salmazole. Number one, this drug is free from CNS side effect. It is true. The log P value is 1.17. This is also true. The 4-AME group was replaced by SO4-SOMe. This is also true. The number D, which says the log P value of the drug is 2.59, is false. No, the precursor or the former the drug which has this RSOMe, this has a log P value of 2.4959, but the salmazole, it has a value of 1.17. That is why the statement number D is false. Now I will go to the next problem. Problem number 10 says contour maps are present in dash. Your options are number A 1D QSR, number B 2D QSR, number C 3D QSR, and number D all of the above. Contour maps are present in which of the following? 1D QSR, 2D QSR, 3D QSR, or all of these three? Let me know A, B, C, or D, which of the following will be the correct answer for problem number 10. Contour maps are present in 1D QSR, 2D QSR, 3D QSR, or all of the three. I will check the chat box after a minute. Okay. Then I will discuss about the contour maps. Contour maps are present in which of the following? 1D QSR, 2D QSR, 3D QSR, or all of the above? Which of the following will be the correct answer? Let me check the chat box. Okay, let's see. Number D, number B, number B. Okay, number B and number D. Two different options I have received. Number B and number D. Okay, let me check which of the following is the correct answer. Number B or number D, or is there any other answer which is correct? The correct answer is actually number C, the 3D QSAR, where the contour maps are present. So the 3D QSAR, or quantitative structure activity relationship, contour maps are the 3D visual representation and used to identify the specific regions around the molecule where the changing its chemical properties, such as adding bulk or changing charge, will likely increase or decrease the biological activity. Unlike the topographic maps that show the elevation, this map used a standard color code to represent molecular interaction field. They act as an activity atlas for the medicinal chemist to rationally design more potent drug candidates. So the common 3D QSA contour maps, these maps are typically generated through the techniques like COMFA, or comparative molecular field analysis, or COMSIA, comparative molecular similarity indices analysis. So the field type steric, the color green, the meaning is favorable region for bulk, action for higher activity at larger or bulky group or methyl or tertiary butyl. The yellow, unfavorable region for bulk, use smaller groups or keep the area empty. Electrostatic, blue region favorable for positive charge and electropositive or electron donating group, and red region favorable for negative and electronegative or electronegative or electron. Hydrophobic, yellow or gray, favorable for lipophilicity and hydrophobic group like alkyl or aromatic ring, and a hydrogen bond purple or cyan, favorable for hydrogen interaction and hydrogen bond donor acceptor, indicated by the specific colors. So how they are created? The alignment, a set of molecules are with known biological activity, are aligned with 3D space, usually based on the common chemical scaffold. Grid probe, a probe or a carbon atom with +1 charge is moved along the 3D grid surrounding the molecule to calculate the interaction of energies at every point. The statistical correlation, the partial least square method is used to correlate the 3D interaction values of the molecule biological activity. The lead optimization, chemists overlay these colored blobs and the lead compound to see exactly where the modifications are needed. Now, for example, a green blob near the specific atom tells the chemist that extending the chain on the position will likely improve the binding. So this is an example of the 3D QSR model. The green portions, they are sterically favored, whereas the yellow portion, they are sterically not favored. The red means the electron rich area, and the blue, it means the electron deficient. Okay. So these are the examples of the 3D QSR and the contour maps. Now I will go to the next problem. Problem number 11 says the sigma m value of the nitro substituent is 0.71, whereas the sigma p value is 0.78. What does the sigma p and sigma m stand for? Number A, electronic effect of the nitro substituent at the meta and para position respectively. Number B, the hydrophobic effect of nitro substitution at meta and para position respectively. Number C, the steric effect of nitro substituent at meta and para position respectively. Number D, the acidic effect of the nitro substituent at the meta and para position respectively. What does the sigma m and sigma m and sigma p values stand for? Number A, the electronic effect of the nitro substituent at the meta para position respectively. Number B, the hydrophobic effect of the nitro substituent at the meta para position respectively. Number C, the steric effect of the nitro substituent at the meta and para position respectively. Number D, the acidic effect of the nitro substituent at the meta and para position respectively. What does this sigma m and sigma p value stand for? Let me know A, B, C or D, which of the following will be the correct answer of problem number 11. What does the sigma p and sigma m value stand for? Sigma n and sigma p values. What kind of effect it denotes? Electronic, hydrophobic, steric or acidic? What does the sigma p and sigma m value stand for? Let me check your answers. Okay, number A, number D. Okay, number A is the given answer for this question, right? Yeah. So I have only received one answer which is saying that number A is the correct answer. Okay. Okay, let me check which if number A is the correct answer or not, or is there any other answer which is correct for our problem number 11. So the correct answer will be very good. It is number A. The sigma M and sigma P, it denotes the electronic factor of this particular substituent at the meta and para position. So electronic factor or electronic sigma factor, the electronic effects of various substituents will clearly have an effect on drug's ionization and polarity. This in turn may have an effect on how easily a drug can pass through the cell membrane or how strongly it can interact with the binding site. It is therefore useful to measure the electronic effect of a substituent. As far as substituent on the aromatic ring are concerned, the measure is known as the Hammett substituent constant. So this sigma is known as the Hammett substituent constant. It is a measure of an electron withdrawing electron coordinating ability of a substituent and has been determined by measuring the dissociation of a series of substituent from benzoic acid compared to the dissociation of the benzoic acid itself. Okay, so benzoic acid is a weak acid and is partially ionizes in the water. Okay, so like this, so the equilibrium is set up between the ionized form and the non-ionized form, whether the relative position of this species is known as the equilibrium dissociation constant. So this is the KH value, which is the concentration of PHCO COO minus divided by PHCOOH, where H signifies that no substituents are here in the aromatic acid. So KH is the value, is the equilibrium dissociation constant, which is the for this only benzoic acid without any substituent. When the substituent is present on the aromatic ring, this equilibrium is affected. The electron withdrawing group, such as nitro group, result in the aromatic ring having the stronger electron withdrawing and stabilizing the influence of the carboxylate ion. And so the equilibrium shift will move to the ionized form. Therefore, the substituent by benzoic acid is stronger acid and has a larger Kx value. Then that substituent X is an electron donating group. The like alkyl group, the aromatic is less able to stabilize the carboxylate ion. The equilibrium shifts to the left, indicating the weaker acid and smaller Kx value. So now for the Hammett constant or sigma x of the particular substituent X, they defined by the following equation. This equation is very important. Okay, that you have to remember. So this is the sigma x is log Kx by Kh or log Kx minus log Kh. Okay, so this is log Kh, log Kx by log Kh. This is the formula that you have to use to calculate the sigma x. Benzoic acids containing the electron withdrawing substituent will have a larger Kx values than benzoic acid itself. Therefore, the value sigma x of an for an electron withdrawing substituent will be positive. Substituents such as the chloro, cyano, CF3 have a positive sigma x value. Benzoic acid containing electron donating substituent will have a smaller Kx value than the benzoic acid itself. Hence, the sigma x value of the electron donatics will be negative. Substituents such as the methyl, ethyl, tertiary butyl have a negative value of sigma. Okay, and the Hammett substituent constant for hydrogen is 0. So this is also important to remember that for this Hammett constant value for hydrogen is 0. The Hammett substituent constant takes into account the both resonance and the inductive effects. Therefore, the values of sigma of a particular substituent will depend on whether the substituent is in meta and para. This is indicated by the M and P by the symbol. For example, for nitro, sigma P is 0.78 and sigma M is 0.71. In the meta position, electron withdrawing power is due to the inductive influence of the substituent, whereas in the para, it is the resonance and inductive both has a play and part. So sigma P have a greater value. So this is also important why sigma P of the nitro group has a greater value than the sigma M, because in case of meta only the electron withdrawing power is because of the inductive effect, but in case of the para, it is both inductive and the resonance which plays a role. That is why the value of sigma P is higher than sigma M. So this is also very important. When once I share the PPT, go through all these things very careful. This is not very much tough. You may feel overwhelmed because of some equations and this kind of all the values, but it is very easy. But you have to go through these things properly, and then you will be able to identify, understand these things more clearly. Okay. Now I will go to the next problem. Problem number 12 says, when two drugs act on a same receptor, the resulting drug interaction is said to be which of the following? Your options are number A predictable, number B unpredictable, number C additive, and number D empty therapy. When two drugs act on a same receptor, the resulting drug interaction is said to have which of the following? Predictable, unpredictable, additive, or empty therapy? Let me know A, B, C, or D. Which of the following will be the correct answer for problem number 12? I will wait for a minute or two and then I will check the chat box. Okay, let me know when two drugs act on the same receptor, the resulting drug interaction is said to be which of the following? Predictable, unpredictable, additive, or empty therapy? When two drugs act on the same receptor, the resulting drug interaction is said to be which of the following? Let me check the chat box. Let me check your answers first. Okay, so today is the last doubt clearing session for me, the regular classes, but tomorrow I will take another class and it will be from 4 to 6 tomorrow. I think you have received, just check once that whether you have received the notification or not. Okay, so tomorrow it is the last class that will happen for this course. I think there won't be any lecture on Saturday, I suppose, because the last lectures will be I mean, whatever lectures are remaining problem solving session that will be over by 15th of the April, which is the Wednesday. So I think there won't be any class on Saturday, but tomorrow there will be a class that I will take from 4 p.m. Okay. The link will be same, the Google Meet link that is you have used to join today's class. So use the same link to join tomorrow's class, but that will be from 4 to 6, not 6 to 8. Okay. Now, I think you will receive the email regarding this particular class. Okay. So number C is the answer that I have received as answer received for this particular problem number 12. Let me check if number C is the correct answer or not. Okay. So the correct answer will be very good. Number C, additive is the effect. Okay. So this is also known as the drug synergy. So when drugs are given in combination, their effects can be antagonistic, subadditive, additive, or synergistic. Drug synergism. So these four terms are very important. One is the antagonistic. I think you know what is the antagonistic because what is the antagonist? I think we have already discussed. This can be subadditive, this can be additive, or this can be synergistic. So drug synergism arises when the therapeutic effect of two or more drugs used in combination is greater than the sum of the drugs administered, individually. So one can think the synergy as inhibiting the drug destroying enzyme. So we have looked in the previously, for example, you have an inhibitor of an enzyme that destroys a drug and destroys the drug, and this will result in synergy. This particular molecule may not have any therapeutic effect on its own, but it only protects the drug from being destroyed. So it is called the sensitizing the drug or synthesizing the pathogen or to the drug, but its primary drug and inhibitors, so the destroying enzymes are administered together, then together it can have a synergistic effect. So we have already seen that for in case of the ampicillin and clavanoid, so they are administered together, so you can protect the ampicillin from the beta lactamase. So I think I have discussed this augmentin, what is the composition for with the ampicillin and clavanoid, and they actually work together and is also known as the this kind of synergic. So we have So with the drug synergy, when the two drugs are given in combination and they can be antagonistic, means that one drug can going to cancel out the effect of the another, and it can be sub-additive. This means there are going to be a little bit of addition to the efficacy, and this can be additive, means the effect of the drug A can be added to the effect of the drug B, and this is going to be result in a combination therapy. But more importantly, what we look at synergy is when the synergy refers to when the therapeutic effect of two drugs used in combination is greater than the sum of the drugs administered individually. So the difference between the additive and synergistic is when I add these two drugs in combination, their efficacy of the net effect does not go up. In case of the additive, but in case of synergy, their efficacy will go up. So so this is the thing that we have these two terms which may seem that they are very, they are similar to each other, but there is a thin line of difference between the additive and synergistic. Okay, antagonist is, you know, that one drug effect of one drug is going to cancel out the effect of another drug, so that is the antagonistic. Subadditive effect of one drug is little bit greater, it increases a little bit with the addition of the second drug. Okay, but when drug A and drug B are given and both are having a effect which is equal to the individual effects of the drug. For example, drug A is producing some effect, drug B is producing some effect. The addition of these two drugs are similar to when these drugs are additive taken together. It is known as the additive effect. But when the drug A has some effect, drug B has some effect, you just combine these two effects, but the effect that is produced by these two drugs when given together is much higher to that one. It is known as the synergistic effect. So that is the difference between the additive and synergistic. These two terms are may seem to be similar, but there is a little difference that you have to take care of or understand. Okay. Now I will go to the next problem. Problem number 13 says, which of the following resistant mechanism describes the function of the beta lactamase? Your options are number A, efflux pump, number B, target mimicry, number C, drug inactivation, and number D, target overproduction. Which of the following resistant mechanisms describes the function of the beta lactamase? Number A, efflux pump, number B, target mimicry, number C, drug inactivation, and number D, target overproduction. Which of the following resistant mechanism describes the function of the beta lactamase? Efflux pump, target mimicry, drug inactivation, or target overproduction? Let me know A, B, C, or D. Which of the following will be the correct resistant mechanism that is described with the function of the beta lactamase? Efflux pump, target mimicry, drug inactivation, or target overproduction? Okay, let me check the answers that I have received for problem number 13. Okay, in case of the antibiotics, I think, see, in case for the antibiotics, you have to mainly remember the classes of the antibiotics. Okay, the mechanisms by which we have actually divided the antibiotics in different classes. So that you have to, you have to remember a few names. If not possible to memorize all the names, but you have to remember some names, right? Okay, so for tomorrow's class in the summary lecture, I mean. Uh, discuss the antibiotics one more time. The briefly I will discuss the antibiotics, okay? Because the best way I think to memorize or to remember the antibiotics is the particular chart or particular figure that is I have already mentioned, which is the antibacterial cell and the mechanisms in the that is mentioned in a single figure. Okay, I will try to discuss about the antibiotics in tomorrow's class one more time. If you say that you are having difficulties, so I hope to give you some kind of summarized way to that you can go through it. Okay. Okay. Number C that I have received for the problem number 13, I guess. So let me check if number C is the correct answer or not. So the correct answer will be yes, number C, drug inactivation. So the primary function of beta-lactamase is to provide the bacterial resistant to the beta-lactam antibiotics such as penicillin, cephalosporin by enzymatically breaking their chemical structure. These enzymes hydrolyze the beta-lactam ring, which is the core part of the antibiotic necessary for the bind to the targets to kill the bacteria. Mechanism of action: beta-lactamases function by making the antibiotics actual target penicillin binding proteins or PBPs, and with the critical difference in how they handle the drug. Binding and acylation: the antibiotic enters to the enzyme's active site. The enzyme attacks the beta-lactam ring, forming a covalent bond by acylation and opens the ring. Hydrolysis and release: unlike PBPs, which stay permanently clogged by the antibiotic, beta-lactamases use the water molecule to quickly break the covalent bond and hydrolysis. Inactivation: the released antibiotic is now chemically altered or hydrolyzed that cannot inhibit the cell wall synthesis, making it completely ineffective. Okay, so this is how the this particular beta lactamase it works actually. So this is the serine beta lactamase, and this is the metallo beta lactamase. So in case of serine beta lactamase, serine is the amino acid that acts on this beta lactam ring, and then lysine we also has here, which also helps in this mechanism. And in the case of the metallo beta lactamase, OH group, the nucleophile is bind with the zinc metal, which attacks and cleaves this thing. The major classes of the beta lactamases are this. We have the categorized by the Amler classification system, that is class A, serine beta lactamase. These are the common penicillinase and extended spectrum of the beta lactamases. For example, these are the examples. Class 2B, metallo like zinc ion break the ring. Then we have serine. Class C, which has the often chromosomal target cephalosporin and cephalomines, and number D, serine oxaloserinase, who are the hydrolyzed carbonapam. Okay. So clinical countermeasure to combat this enzyme, doctor often use beta-lactam inhibitor like clavinic acid or tazobactam in combination with an antibiotic. These inhibitors distract or permanently disable the enzyme, allowing the antibiotic to reach the target and kill the bacterial cell. Okay. So if in today's class it allows if time permits, I will discuss about the antibiotics a little bit. I will give you an overview that if it can be done today itself. I will give you the overview of the antibiotics. From the PPT that I have already shared, you can go through that part, and if it is not clear, I can discuss it in tomorrow's class as well. Let's see if the time is there. I will discuss it in today's class itself a little bit. Okay. I think we will have some time left in today's class. Then I will discuss about the antibiotics. I will give you a brief overview that will help you to, remember these things a little more. Okay, now number 14 number question. It says that the role of the efflux pump in drug resistant is due to number A, metabolizing the drug before reaching the active site. Number B, inactivation of the drug. Number C, decreasing the drug concentration inside the cell. And number D, none of the above. Which of the following will be the correct answer? The correct role of the efflux pump during the drug resistant. Number A, metabolizing the drug before reaching the active site. Number B, inactivation of the drug. Number C, decreasing the drug concentration inside the cell. And number D, none of the above. The role of efflux pump in drug resistant is due to which of the following? A, B, C, or D? Which of the following will be the correct answer? Let me know. The role of efflux pump in drug resistance is due to which of the following? Number A, metabolizing the drug before reaching the active site. Number B, inactivation of drug. Number C, decreasing the drug concentration inside the cell. Number D, none of the above. What will be the correct answer? Let me check the chat box. Let's see if there is any answer for problem number 14. Okay, I have received one answer. Number C is the given answer. Let me check if it is correct or not. So the correct answer is very good. Number C, decreasing the drug concentration inside the cell is the role of the efflux pumps in the drug resistance. So in biology, efflux pump is specialized protein located on the cell membrane that if, actively export the toxic substrates such as the antibiotics out of the cell. These pumps are primarily deliver the multidrug resistant or MDR because they can lower the concentration of the drug inside the cell to sublethal levels. Key characteristic active transport, they require energy to function either from ATP hydrolysis primary or from the electrochemical gradient like proton motive force secondary. Substrate diversity, some are specific to one drug while others are the broad stream, explaining the various classes of antibiotic, detergents and dyes. Structure in gram-positive bacteria, they often form a tripartite complex three-part system and span both inner and outer membrane to eject the toxin directly into the external environment. Major bacterial families, more bacterial efflux pump fall into six categorized superfamilies of the structure and protein source. ABC family, ATP binding cassette uses ATP hydrolysis found in all life form. RND resistant nodulation division powered by proton gradient common in gram-negative bacteria like E. coli. MFS major facilitators superfamily, the largest family utilizes the proton motive force. MATE multidrug and toxic compound extrusion uses sodium and proton gradient. SMR small multidrug resistance, small simple pumps usually providing resistance to lipophilic cations. PACE or proteobacterial, antibacterial, antimicrobial complex efflux, a recently discovered family. So all these things you do not need to memorize, just you have to identify, I mean understand what is the efflux pump. I just have given it for your understanding or for your knowledge. I have mentioned these things. Okay, so what is the biological importance of efflux pump? So beyond antibiotic resistant, this pump play a vital role in bacterial physiology, including the export of the metabolic waste, heavy metal detoxification, cell to cell signaling, quorum sensing. Researchers are currently developing efflux pump inhibitor to use alongside the antibiotics to clog this pump to restore the drug efficacy. So this is also known as the altered drug distribution to the site of action. Efflux pump are transmembrane protein that are often highly expressed by the microorganism and tumor cells to act on the tumor cells that act as a transporters by binding to drugs and carrying them out to the cell before they can exert their therapeutic effects. Some transporters known as the multidrug resistant pump are quite broad in their specificity and can efflux a variety of natural and synthetic drugs. Both specific and broad specificity efflux transporter systems have been identified that contribute to bacterial resistance to a broad range of antibiotic drugs. Okay. Now I will go to the next problem. Problem number 15 says drug resistance of trimethoprim is generated due to number A, efflux pump, number B, sequential blocking, number C, mutation in target, and number D, insufficient drug concentration. Drug resistance of trimethoprim generated due to which of the following? Efflux pump, sequential blocking, mutation of in target, or insufficient drug concentration. Which of the following will be the correct answer of problem number 15? Let me know in chat box A, B, C or D. Drug resistance of trimethoprim is generated due to which of the following? Efflux pump, sequential blocking, mutation in target or insufficient drug concentration. Drug resistance of trimethoprim is generated due to which of the following? Efflux pump, sequential blocking, mutation in target or insufficient drug concentration. Let me check the chat box. Let's see if there is any answer for our problem number 15. Okay, only one answer I have received, which is number B. Others are not answering. I want all of you to answer. It's the last class, so please try and answer the questions. Even if your answer is wrong, it is fine, but try to answer. Okay. Okay, number B I have received as answer. So let me check. The correct answer is at number C, the mutation in target. Okay, so mutation in target is the correct answer. So first, let us see what is this trimethoprim do. So we know this is particular this your the conversion of the pteridine to tetrahydrofolic acid, which is a very important metabolism in the in the bacterial cell or also humans as also. So the pteridine or para-amino benzoic acid, they are converted to dihydroteroic acid by the dihydroteroid synthesis enzyme, which is inhibited by the sulfonamides. Okay, and then this dihydroteroic acid is converted to dihydrofolic acid with the dihydrofolate synthase, and dihydrofolic acid is converted to tetrahydrofolic acid by the dihydrofolate reductase, and this conversion is inhibited by the trimethoprim. Okay. So this is the example of the anti-metabolite. So you have a protein, then mutation can occur here and this result in the conformational change which changes the binding pocket of the enzyme. So this is not a very good for the binding anymore. For example, bacteria resistant to antibiotic trimethoprim derives from a single mutation on the single amino acid of the target enzyme, which is the dihydrofolate reductase. So the properties of a singly mutated enzyme differ from the normal enzyme, and this is still binds to the substrate dihydrofolate. So primary mechanism of the resistance of the trimethoprim basically is the most common ways the bacteria survive trimethoprim by bypassing or alternating this enzyme DHFR enzyme. Acquired DHFR gene or bacteria often acquire endogenous gene via plasmid, transporon or interferon. These genomes produce alternative DHFR enzyme that are naturally resistant to trimethoprim, allowing the cell to continue DNA synthesis even when the drug is present. Target mutation, spontaneous mutation in the native chromosomal FOLA gene can alter this binding site, significantly reducing the trimethoprim's affinity for the enzyme. Overproduction of DHFR mutation of the promoter region of this gene can lead to the overprotection to this sensitive DHFR enzyme, essentially overwhelming that the antibiotics inhibition. Okay. So these are the mechanisms of resistance of trimethoprim. The mutation of the target is the primary mechanism. Okay. Problem number 17, it says aminoglycosides resistant is due to, your options are number A, target mimicry, number B, target overproduction, number C, efflux pump, and number D, lack of the electrical potential gradient across the bacterial membrane. Aminoglycoside resistance is due to which of the following? Number A, target mimicry, number B, target overproduction, number C, efflux pump, and number D, lack of the electrical potential gradient across the bacterial membrane. Which of the following will be the correct answer for problem number 16? A, B, C, or D? Aminoglycoside resistance is due to which of the following? Target mimicry, target overproduction, efflux pump, or lack of the electrical potential gradient across the bacterial membrane. Let me know which of the following you think is the correct answer. I will wait for a minute, then I will check the chat box. Aminoglycoside resistance is due to target mimicry, target overproduction, efflux pump, or lack of the electrical potential gradient across the bacterial membrane. Which of the following will be the correct answer? Let me check the chat box. Okay, number D and number B. I have received two different answers, number D and number B. Let me check which of the following will be the correct answer. So the correct answer will be number D. Lack of an electrical potential gradient across the bacterial membrane is the mechanism of resistance of aminoglycoside. So basically, aminoglycoside antibiotic resistance can arise from the lack of the quinones that mediate the drug transport back and forth from a lack of the electrical potential gradient required to drive the drug access to the bacterial membrane. Aminoglycoside is a medicinal and bacteriological category of traditional gram-negative antibacterial therapeutic agent that inhibit the protein synthesis and contain as a portion of the molecule of an amino-modified glycoside or sugar. Aminoglycoside antibiotics display bacterial activity against the gram-negative aerobes and some anaerobic bacilli that are resistant has not yet arisen, but generally not against gram-positive and anaerobic gram-negative bacteria. So one of the ways by which it can occur by changing the net charge of the membrane. So if you can example, there is a phosphatidylglycerol, membrane that can change to lysyl phosphoroglycyl group. Then it converts to the anionic group to cationic group. This change can affect the permeability of the drug. So if the permeability of the drug goes down, the clarity, the efficacy will go down. In this way, the drug some change can be done, be repelled. So aminoglycoside antibiotic is an example that became ineffective because the quinone that mediate the drug transport from the lack of the quinone of the mediate the drug transport from lack of the electric and potential gradient required to drive the drug across the bacterial membrane. So these two can result in decreased uptake of aminoglycoside, which is going to result in the resistance. Okay, so remember this is very important. Is very important that the lack of the potential gradient, how it affects the resistant the now it generates a drug resistant for the aminoglycosides. Problem number 17, it says that which of the following is the correct reason for the drug resistance of kanamycin? Your options are number A, elimination of the hydroxyl group, number B, phosphorylation of a hydroxyl group, number C, efflux pump, and number D, target overproduction. Which of the following is the correct reason for the drug resistance of kanamycin? Number A, elimination of the hydroxyl group, number B, phosphorylation of a hydroxyl group, number C, efflux pump, and number D, target overproduction. Let me know A, B, C, or D which of the following will be the correct answer of problem number 17. Which of the following is the correct reason for the drug resistance of kanamycin? Elimination of the hydroxyl group, phosphorylation of the hydroxyl group, efflux pump, or target overproduction. Again, I will check the chat box. Check the chat box after a minute. Let me know A, B, C, or D which of the following will be the correct answer. Which of the following is the correct reason of the drug resistance of kanamycin? Number A, elimination of hydroxyl group. Number B, phosphorylation of hydroxyl group. Number C, efflux pump. And number D, target overproduction. Okay, let me check the chat box. Let's see if there is any answer for problem number 17. Okay, no answer for problem number 17. Okay, it's fine. Let me show you the correct answer and let us discuss about it. So basically, in kanamycin, there is the mechanism of resistance is number B, phosphorylation of the hydroxyl group. So this is kanamycin. This is you can see, as you can see, there are several hydroxyl groups are there. Okay, and the phosphorylation of this hydroxyl group makes this particular drug inactive. So another potential approach to countering the resistance to modify the antibiotics in such a way to gain resistance, the mechanism of resistance used against them. For example, kanamycin is an aminoglycoside which is no longer can be used because the bacteria can phosphorylate the hydroxyl groups are present. So because the bacteria can phosphorylate, this is the drug becomes inactive. The active analog can be synthesized, which replaces the susceptible alcohol with ketone. This ketone is in equilibrium with the hydrated gem-diol. When phosphorylation occur, the phosphate group thus formed can is a good leaving group, and the ketone is regenerated. in vitro tests show that the agent was active against the strains of bacteria and was which were resistant to kanamycin. So now this hydroxyl group is phosphorylated, and that is how the particular drug becomes inactive, right? So now if we replace this hydroxyl with a ketone group, the ketone analog of kanamycin, which is with equilibrium with this hydrated gem diol. Okay, and now after the phosphorylation, one hydroxyl group will be phosphorylated, but this phosphorylated group is a very good living group which can easily regenerate our ketone back. Okay, so that is why the phosphorylation mechanism now here it cannot create an effect. Now it cannot make the drug inactive, and this particular analog of the kanamycin, which has a ketone group here in place of the hydroxyl, it is found to be active against some bacteria which are already resistant to kanamycin. Okay. So remember, the mechanism of the drug resistant for kanamycin is the phosphorylation, and to tackle, you have to only remove the hydroxyl group and replace it with a ketone, so that the ketone can form a gem diol. One hydroxyl will be get phosphorylate, but it will also act as a living group and regenerate the ketone. Okay. The problem number 18 says, which of the following mechanism is a major reason for the multi-drug resistant tuberculosis? Your options are number A, target mimicry, number B, target overproduction, number C, mutation, and number D, efflux pump. Which of the following mechanisms is a major reason for the MDR-TB or multi-drug resistant tuberculosis? Target mimicry, target overproduction, mutation, or efflux pump? Which of the following mechanisms is a major reason for the MDR-TB? Target mimicry, target overproduction, mutation, or efflux pump. Again, I will wait for a minute, then I will check the chat box. Let me know A, B, C, or D, which of the following is the correct answer. See, even if your answer is wrong, it is fine. I am saying it couple of times, but try to answer the question. I will check the chat box after a minute, okay? Which of the following mechanism is the major reason for the MDR-TB? Target mimicry, target overproduction, mutation, or efflux pump? What will be the correct answer? Let me check the chat box. Let's see if there is any answer for problem number 18. Okay, I have received one answer, number B. Let's find out if number B is the correct answer or not. So the correct answer is number C. Mutation is the major result for the multi-drug resistant tuberculosis. Okay. So basically, the bacteria multiply at such a rapid rate that there is always a chance to the mutation will render the bacterial cell resistant to a particular agent. This feature has been known for a long time as a reason why patients should complete a full course of the antibacterial treatment, even though the symptom may have disappeared well before the end of the course. This is a rule adhered to with vast majority of the invading because the vast majority of invading bacterial cell will be wiped out, leaving the body own defenses to mop up any isolated survivor or resistant cell. If the treatment is stopped too soon, the body's defense struggle to cope up with the survivors, and any isolated resistant cell is then given a chance to multiply and resulting in a new infection, which will of course be completely resistant to the original drug. This is a major factor of the appearance of MDR-TB. That is why doctors generally say to complete a course of the antibiotics before, although your symptoms may have, have gone, but you have to complete the complete course because there are some bacteria that they stay in the body for a long time, and the more these bacteria, the resist the I mean the bacteria will stay in your body, it will gain the resistance, and if they gain the resistance, they will it will it is very positive that if you take that particular drug in the next time, it won't work, right? The mutation can occur naturally or randomly and do not require the presence of the drug. Indeed, it is likely that the drug-resistant cell is present in the bacterial population even before the drug is encountered. This was demonstrated with the identification of streptomycin-resistant cells from the old culture of E. coli, which had been freezed to prevent the multiplication before the introduction of the streptomycin into the medicine. So remember, mutation is the major reason for this MDR-TB or multi-drug resistant tuberculosis. Now, problem number 19 says, which of the following is used to describe the inhibition of two different enzymes in a same biosynthetic pathway? Your options are number 1, parallel inhibition, number B, sequential blocking, number C, empty therapy, and number D, enzyme inhibition. Which of the following it is used to describe the inhibition of two different enzymes in the same biosynthetic pathway? Parallel inhibition, sequential blocking, empty therapy, or enzyme inhibition? Which term is used to describe the inhibition of two different enzymes in the same biosynthetic pathway? Parallel inhibition, sequential blocking, empty therapy, and enzyme inhibition. Which term is used to describe the inhibition of two different enzymes in the same biosynthetic pathway? Let me know A, B, C, or D. Which of the following will be the correct answer? Parallel inhibition, sequential blocking, empty therapy, or enzyme inhibition? Okay, let me check the chat box. Okay, let me check. Number B, I have received as answer. Okay, let's find out if number B is the correct answer or not. So the correct answer will be yes, it is number B. Sequential blocking is the term that is used to describe the inhibition of two different enzymes in the same biosynthetic pathway. So in biochemistry or in pharmacology, sequential blocking of the enzymes, also known as a sequential inhibition, is a strategy where two or more inhibitors act on a different steps of the same metabolic pathway. This approach is often results in the synergistic effect, which means the combined impact of the significantly greater than the inhibition, even either inhibitor was used alone. How sequential blocking use? Metabolic pathways are often linear chain when the product of the one enzyme becomes the substrate of the next. For example, A to B is the first enzyme works on this conversion of A to B, from B to C is the second enzyme, and from C to D is the third enzyme. In a sequence, if an inhibitor blocks E and another blocks E1 and another blocks E2, the pathway is blocked at the two sequential points. This is highly effective because reduced leakage. The first inhibitor is not 100% efficient. Some substrate will still pass through, and second inhibitor acts as a backup to catch the remain. Synergy, the reduction of the intermediate B makes the second enzyme even more vulnerable to the inhibitor, so the less substrate is complete to the inactive site. For example, now the examples like clotrimazole, as mentioned, the gold standard example for this sequential blocking is the clotrimazole. One first drug is the sulfamethotrexate, which blocks this dihydrofolase synthase, and the trimethoprim blocks the dihydrofolase reductase by hitting the two-step in combination is bactericidal, whereas the each drug alone is the bacteriostatic. Okay. Then sequential nephron blockage or CANSNB used in patient with a diuretic resistant, often in severe heart failure. This strategy blocks the sodium reabsorption at the segment of the kidney's filtration unit from nephrons. So drug 1 is a loop dehydrate like furosemide, which block the sodium transport to the thick ascending limb, and drug 2 is the thiazide, like metronidazole, which block the sodium at the distal convoluted tubule. The result, this prevents the kidney from compensating the first drug, reabsorbing the salt and the line, leading to the massive fluid clearance. Then dual RAS blockage in the renin-angiotensin-aldosterone system regulates the blood pressure. Doctors sometimes use two different sequential point. One is the SC inhibitor to prevent the conversion of angiotensin 1 to 2, and ARB, okay, that's receptor blockers which are like closartan, which block the receptors of angiotensin 2 actually works. Note that while biochemically sound, this is cautiously to used today because it carries high risk of kidney strain and high potassium. And cancer chemotherapy also the sequential blocking is used, for example, with methotrexate and 5-fluorouracil, where the methotrexate depletes the folate pools and the 5-fluorouracil is more easily inhibits the thymidine synthesis because the cell is already primed by the lack of the folate. In the central insulin protocol, these drugs to create a choke point for the DNA synthesis. So these are the examples where the sequential blockings are used in the different therapeutic areas. Okay. Now, the last question of today's session, it says, which of the following is an inhibitor of inosine 5' monophosphate dehydrogenase? Your options are number A, dopamine, number B, mesoribin, number C, propanolol, number C, ciprofloxacin. Which of the following is an inhibitor of the inosine 5' monophosphate dehydrogenase? Dopamine, mesoribin, propanolol, or ciprofloxacin? Let me know A, B, C, or D. Which of the following will be the correct answer? Which of the following is an inhibitor of inosine 5' monophosphate dehydrogenase? Dopamine, mesoribin, propanolol, or ciprofloxacin? Let me know A, B, C, or D. Which of the following will be the correct answer? Okay, let me check the chat box now. Okay, I have received two answers. One is saying number A, another is saying number B. Let us find out which of the following will be the correct answer, number A or number B. So the correct answer will be number B, mesoribin. Okay, it is the inhibitor of the inosine 5' monophosphate dehydrogenase. So this is the structure of mesoribin. So it is also known as the bradykinin, an immunosuppressive drug primarily used in Japan and other parts of Asia. This is a nucleoside of the imidazole class that is valued for specific mechanism of action that targets the immune system while being generally less toxic than the other traditional immunosuppressant like azathioprine. In the mechanism of action, it is an anti-metabolite that inhibits the de novo purine synthesis pathway, which is essential for the DNA and RNA production. The target enzyme, it selectively inhibits the inosine monophosphate dehydrogenase or IMPDH or guanosine monophosphate GMP synthase. The selective inhibition by because lymphocytes like T B cell rely almost exclusively on the de novo pathway of proliferation. Other cells that use salvage pathway, mesoribin specifically suppresses immune cell multiplication without significantly harming the other tissues. Cell cycle arrest, it arrests the cell cycle space, effectively stopping the immune response at the point of the cellular division. Clinical uses, mesoribin is frequently used as a part of the combination therapy with the corticosteroids for the several condition organ transplantation, primarily used to prevent the rejection of the renal transplantation, renal diseases, is effective to the lupus nephrosis, the nephropathy and nephrotic syndrome, and rheumatoid disease like arthritis. It is also used in other autoimmune condition. Comparison with other drug, it is also compared to the mycophenolate, mofetil and azathioprine. Okay, so safety profile compared to MMF, the mesoribin has a lower risk of gastrointestinal issues like diarrhea and viral infections such as the CMV. Toxicity, unlike azathioprine, this is not incorporated into the cellular nucleic acid, which is results in less hepatotoxicity or liver damage and myelosuppression or bone marrow suppression. And notable side effect, the most significant drawback is the higher risk of the hyperuricemia or the uric acid level, which can lead to gall or the kidney stone. Okay. So this was the last question of today's session. So basically today we have discussed majorly about the various mechanisms of the drug resistant, and also we have discussed about the QSAR. One doubt that has been, that is raised because for the antibacterial agents, so how to remember. So I think, see, the antibacterial agent, I know it is a very difficult to remember the vast range of the antibacterial agents, but the way I actually prefer to remember this is the remembering the figure. Just let me. So this is the PPT for the week 10 live session. I think you know how to where to find out this PPT. Now you have to only go to the problem solving session materials, and you can in the Google Drive you will find this PPT. Okay, so this particular, just no, this not this one. Just let me show you the figure. Okay, this figure actually I prefer generally this figure, this problem number 16, you will find this figure. This I actually prefer because here you can see the all the things are very well, mentioned, that the mechanisms of the various antibacterial agents and to remember. So this is a bacterial cell. We have a cell membrane. Okay, so on the cell membrane, you have to remember that there are three types of, majorly three types of reagent. Cell wall synthesis inhibitors, nucleic acid synthesis inhibitor, and protein synthesis inhibitors. Okay. So cell wall synthesis inhibitors like beta-lactam, anti-beta-lactams, like for example, penicillin, cephalosporin, carbamazepam, monobactam. Vancomycin or bactricin are also there, and in case of cell membrane, polymyxins are the reagents. Okay. Now comes for the nucleic acid synthesis. So if we have the folic synthesis, these are the anti-metabolites like sulfonamide and trimethoprim that I have already mentioned today. The conversion of PABA to DHFA and THFA, that is inhibited by the sulfonamide and trimethoprim. Together they are used as also for sequential blocking, and the DNA guidage are inhibited by the quinolone class, and for DNA polymerase by the rifampicin. In case of the protein synthesis, in the ribosome, 50S units are inhibited by the macrolide classes, the clindamycin, linazoloid, chloramphenicol of streptogenesis, and 30C are the tetracycline aminoglycoside. Now, remembering the names, I think in the classes, different classes have a several name. For example, in the macrolide are the erythromycin, the macrolide erythromycin, telithromycin. So, whenever there is a mycin, you have to remember that it is a macrolide class of the antibiotic. Okay. For example, this is also important. This problem is also important. Okay, so the different mechanisms. So in case of the antibacterial agent, when you are reading about the antibacterial agent, they may seem to like lots of the drugs are there that you have to remember the lots of them. But the easiest way to remember is the remembering them by the mechanism of action. Like the classes of drugs that are the involved in the cell metabolism, the drugs which are the involved in the inhibition of the cell wall synthesis, the plasma membrane, then protein and nucleic acid synthesis. If you can remember, so the figure is the, I think I found this figure is the most easiest way to remember this particular names of the drugs. Okay, so you can go through these slides, and if you still if I feel that it is not easy to remember, you can let me know tomorrow, then we can figure out together something. Okay. And I think you have, I hope all of you have downloaded the books that I have mentioned, that medicinal chemistry. I will also write the name of the book. Okay. The mm, medicinal chemistry book by Graham and L. Patrick. Okay. The name of the book is The Introduction of Medicinal Chemistry. Intro. You can go through the book. It is a very good book actually for your course. It is a perfect book. You can go through this book. The PDF is available. If you cannot find the PDF, you let me know. I will, via email, I will share the PDF of this particular book. So tomorrow, basically, so whatever today I have mentioned regarding the antibiotic resistant, if you go through the particular slide, you will find already all the mechanism I have mentioned today. Like, so these are the major classes of mechanism, and also the particular slide that course the I think the 10th PPT, you will find some mechanisms are there. So there is, I think, nothing left for discussing in this case of this resistant. Tomorrow, I have planned to discuss some antibiotics, and also I have planned to discuss some antiviral agents that I couldn't discuss in earlier. Okay, so let's see whatever I can do for tomorrow's class. I will majorly focus actually on some antibiotics and on some antiviral and some anti-cancer therapeutics. Uh, yeah, I will try to discuss whatever is left actually for discussing, and don't I mean feel scared with the examination and all the course load, because because all the things I have already mentioned in your class, and all the things are mentioned in the PPT. Okay, the major important core topics all you will find in the presentation if you go through the materials whatever is shared to you here in this Google. So I think you know where to find this problem solving session in the SWAM portal. If you go to the SWAM portal, at the end you will find the problem solving session recording. Clicking on that, you will find a presentation link. And here, this Google Drive, you will find all the presentations. Okay, go through the presentation in each and every class. Actually, I try to discuss all the important things and pinpointing this. Whatever is left, I will discuss tomorrow, and I will try to give you a little overview about the antibacterial just because you have asked. I will try to include a PPT or a slide with discussing the antibacterial agents one more time. And basically, it is just I have mentioned that remembering the easiest way to remember that thing is remembering the particular figure. Okay. So thank you all for today. If there is any doubt or query, please feel free to reach me out via this email ID. Okay, and try to yeah, I will upload today's slide and the recording after at tonight by tonight or by tomorrow morning. You will find the presentation recording and also the slide. Okay. Thank you all for joining. Thank you all for answering and interacting. Let's meet tomorrow for our last class from 4 p.m. Okay, so use this link for joining tomorrow's class. We will discuss, we will having our final class tomorrow. So please try to join. Thank you all. I will leave the session. I will end the session for today. Thank you again.